Okay, so it's a real pleasure for me to be able to introduce our BioPhorum webinar on an industry perspective on the use of force degradation studies to assess comparability of biopharmaceuticals. BioPhorum is a global collaboration, and we connect the biopharmaceutical industry to basically solve problems and challenges and work together to do process development faster and more efficiently than maybe you would be by yourselves. It's fantastic to see different approaches to problems, and it's in that spirit we have our collaboration. We have 11 forums that span the whole of biopharmaceutical development, and the development group is one of those forums. We have 35 member companies in the collaboration, and they collaborate across 13 work streams. There's around 1,000 active participants that contribute to all of these. We do benchmarking surveys, we have general discussions, but we're very driven by what our members want to talk about. The force degradation work stream is one of these work streams, and that began in 2015. And that paper did much work and benchmarking surveys, discussions that led to the paper that we're going to talk about today. And I'm just going to ask my presenters now to put on their webcams so you can see them all. Fantastic, I can see them all appearing now on the screen. So we have John Campbell from GlaxoSmithKline. We have Jamie Doyle from Regeneron, Goeran Huebner from Boehringer Ingelheim, Kasia Nowinski from Seagen, now Pfizer, Alex Parvon from Merck & Co. Incorporated, and Surinder Singh from BMS. And I work for BioPhorum. If you're interested, our paper was accepted for publication in the Journal of Pharmaceutical Science as a commentary, and that has appeared online and in libraries around the world in the last week or so. So we're very excited about that. So what we're going to be talking to you today is we're going to give you an introduction to comparability and how force degradation can be used for that. And we're going to look at factors that influence force degradation studies. How do you design those studies? We're going to cover that as well. And your analytical characterization and testing strategy is really important to understand what to do for that. This will all culminate in evaluation criteria, and that's where we really consider what you can do with the data. We also look at how you can apply this to non-platform biologics, and we'll finish up with some conclusions and then a Q&A session. So I'm now going to hand over to Kasia, who is going to introduce comparability. Kasia.
Thank you, Elaine. Hi, I'm Kasia Nowinski from Pfizer, located in Bothell, Washington, and today we'll start off with a brief introduction to our topic. We'll be talking about comparability assessments. Comparability assessments are needed when there are changes made to your drug manufacturing process to ensure there's no adverse impact on the quality, safety, or efficacy of your drug product. Comparability assessments can have a number of different components as part of them, shown by some of the activities listed in the green circle, including batch testing, release testing, stability data, extended characterization, or non-clinical and clinical studies. Force degradation studies, on the other hand, apply stress conditions to a drug substance or drug product, which can exceed those used for stability studies performed according to ICH Q5E. They have a number of different applications, as shown in the blue circle, including formulation development, method development, identification of critical quality attributes, structure-function relationships, and others. Today, our talk really focuses on the intersection of these two topics, and specifically when comparability is applied in forced degradation. Our force degradation work stream recognized that there is limited guidance on how to design and interpret force degradation studies. ICH Q5E explicitly avoids the prescription of a particular comparability strategy. However, many companies conduct risk assessments which help guide their strategy for comparability as well as the scope of testing. These risk assessments can be influenced by the nature of the manufacturing change that's occurring, the potential for that change to impact quality attributes, the availability of suitable analytical techniques, the phase of development, and relevant clinical and non-clinical data. ICH Q5E does cite that accelerated and stress stability studies or force degradation studies can be useful tools to establish degradation profiles and provide a comparison of your pre-change and post-change product, but the details around how to set up these studies are limited. Next slide, please. Our force degradation work stream conducted a survey to probe the industry practices on when and how force degradation studies are used in comparability assessments. Each survey question received responses from at least 14 different pharmaceutical companies, and each company was limited to a single response. The survey focused on factors that influence the decision to include force degradation in a comparability assessment, how to design those studies, the analytical testing strategies, how to evaluate the data, and the application of force degradation to non-MAB modalities. Overall, we found that force degradation studies are used by all companies to support comparability. However, a force degradation study isn't always appropriate or needed for every single comparability assessment. When the studies are performed, the same process is used across companies, and that's illustrated by the flow chart on the left. First, the force degradation study is performed on material from before and after your manufacturing change, referred to as pre and post-change material. A study protocol is developed. The samples are then all stressed. Analytical characterization and testing is performed. The data is analyzed, and then a study conclusion is drawn on whether your pre and post-change material are comparable or not. If the answer is yes, that data is likely included in a submission to health authorities to support your overall comparability assessment. And if not, then typically the results need to be reanalyzed or confirmed, or additional data may need to be collected. And now I'll hand it off to Goeran, who's going to cover our next topic on the factors that influence the use of force degradation studies.
Kasia, thanks a lot for the great introduction to the topic. I'm Goeran Huebner from Boehringer Ingelheim, located in southern Germany. And now I would like to continue and outline a couple of considerations for using FDS to support comparability. Respondents to the BioPhorum survey indicated that force degradation studies are used by all companies to support comparability. However, an FDS study is not always appropriate or needed for every comparability study. The design and extent of FDS are driven by various factors. The extent of changes in the manufacturing process is a key driver in deciding whether to implement FDS as part of the comparability assessment. A common approach among all responding companies is to use risk-based assessments to rank process changes on the amount of perceived risk into categories such as major, moderate, or minor. Major process changes may suggest the addition of FDS to a comparability study protocol. The size and scope of the FDS increases with increasing perceived risk to product quality. Examples of major risk might include cell line changes for drug substance, or, for example, formulation composition changes for drug product. Whether the manufacturing change occurs at the drug substance or drug product process step is not necessarily a driver of whether or not FDS is needed, although it can influence what material the FDS is performed on. If the major change occurs at the drug substance process step, FDS is performed on drug substance material. If the change occurs at drug product, FDS is performed on drug product. However, sometimes FDS is done on drug product even when changes are limited to drug substance. All risk-based assessments or risk-based assessment is heavily influenced by prior process knowledge, including individual company experience with that modality or product. Next slide, please. Another consideration for determining the need for FDS is the phase of development for which a product may require a comparability study. In contrast to late phase development or post-approval, in early phase development, a less extensive comparability exercise or comparability study without FDS may be sufficient for the evaluation of comparability. However, early phase development work may trigger FDS if there is a need due to a major process change. Part of differences in need for data is the expected amount of clinical experience that is to come. Earlier phase trials are still refining processes and have opportunity to see impact to patients in a clinical setting, while therapeutics in later or post-commercial space are seeking either proof of concept or an expected outcome for a defined product. This position is consistent with ICH Q5E. Overall, FDS to support comparability occurs most often during late phase development. More frequently, major process changes are observed during later phase development, with phase 3 processes typically being representative of the final commercial process. While the phase of development may influence the risk-based assessment, with later phase increasing risk in some instances, the amount of risk, so major, moderate, or minor, along with the amount of available pre- and post-change stability data expected at the time of filing, are the key drivers for determining the need for FDS. In general, less stability data, so both long-term and accelerated conditions, available at the time of filing mean that there's more risk associated with comparability with respect to the degradation pathways. Finally, at the commercial stage, there may be sufficient data from existing stability to support comparability. However, the decision to omit FDS from the comparability study should also at this phase be based on a risk assessment, and in most cases, comparability studies in the commercial phase are as comprehensive as in the late stage development. I would now like to hand over to Jamie Doyle from Regeneron, who will present more details on the force degradation study design. Thank you.
Thank you, Goeran. Yep. So now that we've kind of introduced the need for a force degradation study based on the risk assessment and various factors that Kasia and Goeran have discussed, we'll talk about how to actually set up your force degradation study. So there's a couple factors you'll have to consider. This will be CQAs, the critical quality attributes of your molecule, and what changes you're looking to get out of your material. And then what conditions specifically you can apply that will prompt that change in your material to target a specific CQA if that's your goal. You'll also want to look at prior knowledge that you have from previous studies. Is there any data that you've already gathered that you'd be able to leverage to potentially modify a condition? Maybe you need something more or less aggressive, or remove a condition altogether because you may already have pre-existing data to satisfy the risk that you're trying to address. You also may want to look at the data that you have to see if there are any additional gaps that you'd like to fill by leveraging your force degradation study, and you can build that into your robustness of the data that you'll present and move forward with. You'll also want to look at the stress conditions more closely to figure out what you could use to induce a specific type of degradation profile within your molecule. You may also want to consider a range of conditions if you haven't already looked at conditions previously, again, looking for a gap. and also the time points. So some conditions may be more or less aggressive, where you may want a shorter study so that you don't completely obliterate the material, or you may want a longer study to try to optimize the degradation of the molecule overall. And this will include looking at what phase your material is in, and also the length or amount of conditions you test may have some limitations in terms of the batches that are available to you in material quantity. Once you go through your kind of targets, you'll actually look at applying specific stress conditions. So if we move into the next slide, Elaine. There's several different stress conditions we can look at, and you can see the respondents from, or the responses from the respondents in the plot in the bottom right. Thermal stress seems to be the most prevalent. This is most likely because it's one of the easier stresses to induce degradation within your molecule. And there's multiple different reasons we would look into thermal stress. And you can generally look at a range between 40 and 55 degrees C, which is what was provided from our respondents as the typical temperatures applied. You'll notice that these tend to be a more aggressive condition than what's used in standard accelerated stability. And that is because we are trying to target degradation of the molecule for the study. Although temperature stress alone may not be sufficient to induce the changes you're looking for, specifically if you're trying to target very specific CQA within your molecule, you may want to consider other conditions or a combination of conditions. These can include photostress, oxidative stress, several of the other conditions that you see in the plot. And there's more information on these actually in a sister publication that we have from an industry perspective on biopharmaceuticals. This was a previous publication, I believe, from 2020 that goes into a bit more of the stress conditions that are commonly used in forced degradation studies as well. So once we've highlighted what stress conditions we want to use, we do have to consider the time points for the study so that we don't pull too many or too few. Stress duration will depend on how aggressive the stress is. Typically, our respondents will target less than a month for our stress, but there can be a range. You may pull more or less, 3, 4, 5 different time points within the study, as you can see from the plot on the right. So how we have this broken out is to show the diamonds that you see are individual time points that respondents had replied with. So just 2 time points used, 3 time points used. And the bars that you see are a range of time points that respondents have come back with. So depending on the stress condition and the study, a lot of respondents agreed that a range may be applicable. And this could be different ranges also for different stresses. But the majority of respondents came back with 3 to 5 time points being the preferred choice. So once you've identified the time points and the stresses you're going to apply, you need to consider what batches and the number of batches that you're going to look at. Ideally, from a statistical standpoint, we'd prefer 3, and you can see that as one of the most common responses from the respondents. However, you may not always have 3 batches available. So there could be some limitations on either your pre or post change material availability, hence why also there's a difference in the number from pre-change to post-change. So 3 is preferred, most likely for statistical purposes, but again, that's not always possible depending on material availability and also your phase of development. So you can see 3 or less seems to be the most prevalent response from our respondents to the survey, but there are areas in availability that can allow for 4 or even 5 or more, which would be fantastic, but unfortunately not always possible as we're going through the comparability assessment. So after that brief walk through our setup for this study, we're going to talk about the analytical characterization and testing, and Surinder is going to lead us through that.
Okay, thank you, Jamie, and hello, everyone. I'm Surinder from Bristol Myers Squibb, located at New Brunswick in New Jersey. And after discussing the survey results about how to create FDS, all these samples, the next set of questions we surveyed at the forum is about the analytical characterization strategy and what are the different tests. Elaine, can we go to the next slide, please? Yeah. So basically, what kind of tests should be included in the analytical characterization, and what are the drivers those dictate inclusion or exclusion of certain specific tests? So those were the questions those were asked in the survey, and the results showed that the primary driver that dictates the inclusion of these tests in the characterization strategy is available knowledge about the degradation mechanism of the molecule of the interest in the comparability study. And in addition to that, there are some other factors those influence the characterization strategy, like development stage of the project, then nature of the process change, or nature of the material, is it DS or DP, or the impact of change on the product quality, safety, and efficacy in the light of CQA risk assessment. And there are some other method-later considerations like analytical variability and testing efficiency. And we will speak to these factors and considerations in the next slide in a little bit more detail. So here in this slide, these are the survey responses and describing various considerations and factors those influence the analytical characterization strategy. For example, the phase-dependent considerations. If the program is in early stage of development, then it may not be necessary to perform extensive extended characterization on FD samples. However, if the program is in the late stage because the risk associated is high, then it adds value to perform extended characterization on FD samples as well. The next is the nature of the material. If the change is at the DS level, then it may not be necessary to include subvisible particle analysis, as particles can be cleared when DS is getting converted to DP. And likewise, if there is some formulation change that is happening at DP level, then as we do not expect much of the impact on the N-glycan profile of a molecule due to formulation changes, and therefore it may not be necessary to perform N-glycan analysis on FD samples. The third one is assay selection. The focus should be on the stability indicating assay and in correlation with the kind of FD condition that was used to generate these samples. For example, if there is some oxidation site in the CDR and H2O2 treatment was used, then it makes sense to include tryptic peptide mapping or HIC chromatography to quantitate the relative abundance of oxidized species in post-change material when compared to the pre-change material. Then there are some additional considerations. If we see some unusual results, for example, there is some elevated levels of HMW species in the post-change material, then it adds value to include some other orthogonal techniques like SAC-MOLs or analytical centrifugation to get better understanding about the molecular weights of the species, so that we can have a better insight into the risk due to HMW species, because there is a perceived risk that higher HMW, especially higher order HMWs, they can cause immunogenicity and things like that. Then we have timelines as a factor. If the timelines are very short, then probably, even though we generated samples for 3 months, but we don't have enough time to include those. So 2-month time points should be good enough. We have enough time points in between to get the idea about the trend, how the molecule is degrading, and that way we can save times to meet the filing timelines. Then there are other conditions, those are pertaining to method quality attributes like analytical variability. If the method has high analytical variability like cell-based bioassay, then it makes sense to perform this assay on the samples, all the samples for that particular condition in one go instead of performing on different days. That way we can minimize the variability and we can have more confidence in the trends. And we have then another method-related attribute like testing efficiency. Some methods are very low throughput like tryptic peptide mapping or cell-based bioassay. So instead of including these tests for all the samples, or we can begin with just initial and the final time points. And if there is not much difference, then probably there's no need to include in between samples. And if we see a difference, then we can include in between later. So these are all the factors and the considerations. And the next set of questions we asked about is, what are the assays those are typically included in the characterization strategy we perform on all these FD samples. And as you can see in this slide on the top green box, antigen binding, charge variant distribution, and size variant analysis. These 3 attributes are the most commonly tested on FD samples. As you can see, 18 out of the 18 respondents are performing these tests on FD samples. The next in the tier are peptide mapping, cell-based assay, and concentration. Those are also frequently used. You can see that 12 to 14 respondents out of the 18 are testing their FD samples with these methods. And then there is a list of others. I don't want to go in the detail of each, but in the interest of time, but their inclusion is based on the considerations, the factors that we discussed earlier in the previous slide. For example, if there is a change in the protein concentration from low to very high, say 20 going to 150 or 180 milligram per ml in DS, then probably it makes sense to perform appearance, subvisible particle analysis, and higher order structure analysis on FT samples. Similarly, if there is some change in the formulation composition that can impact the deamidation or let's say oxidation in the FC region, then it makes sense to include FC receptor binding analysis on FD samples. There can be some other changes in the formulations, like buffering agent is being by something else, or we are adding or replacing surfactant and antioxidizer elements in the formulation. Those can also impact the overall stability, and that's why sometimes even pH, excipient content, and turbidity, and some other attributes can also be included in the analytical characterization strategy when it comes to FD samples. So once we perform all these tests and we have all the results available, the next question is about how to make sense of it, and my colleague John will speak to those in greater details. So all yours, John.
Thank you, Surinder. So as Surinder mentioned, I'm John Campbell. I'm with GSK in the Philadelphia area in the United States, and I'm happy to go through this next section on evaluation criteria for force degradation when it's used in comparability. So once all of the force degradation samples have been prepared and then analyzed, the next step is to compile all that data and evaluate it to assess comparability of your pre and post change. Now, those familiar with Q5E will recognize the quote here, where the expectation in Q5E is to have predefined criteria that is set before the study is conducted, against which the results obtained for the pre and post change material will be compared. So one of our objectives in this survey was to determine how companies are handling this. Now, the vast majority of survey respondents, both in the survey responses and in conversations held regarding the survey, do not define quantitative pass-fail acceptance criteria. In fact, only one company stated that they did. So there's no numerical pass-fail criteria, and most felt that was very challenging to set, and so that's not something that is recommended. However, what is done is it's predefined how the data will be evaluated. So not necessarily pass-fail criteria, but defining how the data will be taken and what aspects of the data will be looked at, and that includes a comparison of the degradation pathways as well as the degradation rates between the pre and post-change material. In terms of the things done to facilitate that evaluation, you can see the question on the right here. How do you evaluate your studies? And we had 18 survey respondents, and it was kind of a mix of visual plots, review of the raw data, small number of companies reporting using statistical evaluation, and then quite a few suggesting that they use some combination of all three of those to evaluate the data. So on the next slide, there was some interest expressed in understanding how to use statistical evaluations to understand the data better. Unfortunately, many of these studies don't have a data set large enough to conduct a really robust statistical evaluation. Now, the person who's preparing all of the force degradation samples and the individuals testing those samples may not agree with that, but a statistician looking at the results would say that the data set is not actually very large for a really robust statistical evaluation. However, companies mentioned that t-testing as one common strategy being employed when doing that. There are other options for ascertaining whether the differences seen between material are statistically significant. But again, that proves challenging in many cases. And so when a numerical statistical evaluation cannot be completed, which is often the case, the approach involves just a comparison of degradation rates, visual comparison of plots, and maybe alternative assessments as well. Go to the next slide. So just to kind of illustrate how this might be done, on this slide is some hypothetical size exclusion chromatography data. And plot 1 on the lower left there, you see we've got 6 batches of material being analyzed. The high molecular weight percentage is assessed over the course of several time points, several days of stress. We've got 6 batches, 3 of which are from the pre-change process, 3 of which are from the post-change process. And what we're showing in plot 1 is something that we would consider, a result that we would consider supportive of comparability, where there isn't really a clear difference in between batches. And so because there's no meaningful differences, this component of the study would be considered supportive of comparability. However, you can contrast that with plot 2 on the right, where we see a clear difference. Despite some analytical variability, we see the post-change is consistently degrading at a faster rate than the pre-change material, despite being presumably stressed side by side in the exact same conditions. And so in this case, an additional evaluation should be performed to understand what's leading to that difference observed, and whether those differences can be justified in a scientifically appropriate manner, and then their subsequent impact on the comparability conclusion. And obviously, this assessment needs to be formed not only for SEC, but also the other analytical techniques that were applied for the study. After reviewing the survey results and participating in the discussions, we did our best to put together a kind of flow diagram for decision making when you're looking at the data in terms of getting to possible conclusions. So, I mean, the first step is, once you've compiled all the data and compared it, is do we see differences between pre and post change material? If the answer is no, meaning we can't distinguish any difference in degradation rates and degradation pathways, we slide down the left side of this diagram and we conclude this study is supportive of comparability between the pre and post change material. If we do see differences, and that would be unexpected, and again, differences in degradation rates or pathways, the first step is actually to kind of rule out any sort of experimental variability contributing to some arbitrary, not real difference observed between the processes, or an analytical artifact or something like that. And this is one case where statistics can come in if desired, just to determine if those differences are meaningful. So if the differences can be attributed to just analytical variability or some artifact in the testing, then again you go down the left and say, yeah, we don't have any meaningful differences observed, and the study is considered supportive of comparability. And if you conclude that the differences are real and are meaningful, there are a couple of options. One on the left is actually if you have superior stability, at least in the forced degradation study, of the post-change material, this would not be expected to pose any risk in terms of long-term stability or safety or efficacy for the patients. It would actually be a process improvement. And so would likely need no further justification before you conclude, yeah, the study is comparable, although you may want to understand what is leading to the superior stability of the post-change material and what led to that observation. Then we come to the alternative scenario on the right, which is inferior stability of the post-change material in the forced degradation study. And in this case, the differences are meaningful. The forced degradation study would not be supportive of comparability. And so some additional work would be necessary here to assess the risk and impact to the overarching comparability study. Most companies discussed needing to plan for further work to understand what the differences are and what's leading to those differences. And then once that's complete, performing a holistic review of the data and any prior knowledge to ascertain if there's any risks to patients' safety and efficacy, and again, the overarching comparability conclusions. So it's important to remember that when determining comparability, forced degradation is not the only component of that. And so the comparability conclusion isn't going to hinge on that alone, but it needs to be taken into context with the other components of the study, such as GMP release testing, extended characterization, potentially long-term stability data, things like that before drawing an overall conclusion. So it's really about, does the forced degradation support that or not when ascertaining and evaluating the data. And also, we'll point out, remembering from Q5E, that comparable doesn't mean identical, but highly similar. So really, it's when you see differences, all is not lost. It's just a matter of understanding what those differences are and ensuring that there's no adverse impact to safety or efficacy expected. So I think that brings me to the end of my section. And next, Alex will pick up talking about non-platform biologics. Alex, so over to you.
Thank you, John. So maybe to close out, I'll just cover certain aspects or considerations that the survey intended to also address around non-platform biologics. So if you go to the next slide, and my name, by the way, is Alex Pavon, and I work at Merck and in Kenilworth, New Jersey. So what do we consider non-platform biologics? So one of the things that we intended to achieve is, and through the survey, is to understand how these modalities are defined, and in general, non-platform biologics are anything, outside of the monoclonal antibody, the particular isotypes listed here, IgG1, 2, and 4. And the reason for that is because most companies, in fact, I would say all the companies that were part of the survey, expressed that they don't have as much manufacturing experience with these modalities relative to with platform monoclonal antibodies. So I guess lack of experience, lack of just enough experience working with them, as far as, not just the analytical methods, but as far as just understanding the modes of degradation is what is kind of led to the classification of what we consider non-platform. So what are those? So what the cartoons shown on the left here are depicting are what we consider a heterodimer, which will be a highly glycosylated protein with an FC domain. Or likewise, it could be something, or similarly, I would say, something like a fusion homodimer protein, also highly glycosylated, it could be N-oral glycan. Or we can also put in this category of other types of fusion proteins, insulins, I would say other bioconjugates that are linked to PEGs or domains that really will fall out of that category of your typical monoclonal antibody. On the bottom here, what you see here are what we consider also non-platform. While they do have the monoclonal antibody component as far from the biologic side, they also are attached to a drug linker. So this is what we call actually obviously an antibody drug conjugate, and we can subdivide those into antibody drug conjugates that are non-cleavable and cleavable, and that will really depend on the chemistry of the linker, whether it's enzymatically cleavable or not. So that also poses additional challenges around really how do we do studies for these modalities? What are the type of analytical tools that we implement? I would say that just based on the information that we have, in the manuscript, I would say, putting a plug for everyone to kind of look at it or read it, is that the principles that we describe here in the publication from 2020 broadly apply, but there's some adaptation that will have to be implemented considering the difference of physiochemical properties. The other thing to consider is also the analytical testing panel. Given the complexity and the additional attributes, you may have to do sample manipulation or treatment to really probe for the degradations, for example, for the fusion proteins on the polypeptide, and to sort of unmask what those changes are, because for a lot of methods, the glycosylation pattern typically tends to dominate or obscure the modes of separation. So and as an example, that would be utilizing the glycosylating in a fusion protein to kind of monitor the char heterogeneity coming from the polypeptide. For antibody drug conjugates, that will entail obviously testing for the drug to antibody ratio, as well as the levels of free drug, toxin, and the degradation pathways that may arise from these stress conditions. Next slide, please. So what do we mean by adaptation? So typically, initially, we have to perform some what we call scouting studies. So that will be identify the suitable conditions that we make sure that we're generating levels of degradation that are suitable to make meaningful conclusions out of the data and not completely degrade the molecule, and obviously not to stress them enough where the impurity levels that you generate are not sufficient to sort of you generate enough data to claim comparability. So that for two examples that are shown here on the left is really maybe the way you would modify or adapt high pH, low pH, as well as the thermal stress. Thermal stress, for example, in the guidance document that we published back in 2020, there was some best practices on how the conditions are utilized for monoclonal antibodies. In this case, those conditions may or may not apply. In particular, for example, when we're talking about the thermal stress, If your melting temperature is lower, you don't want to stress your molecule to 50 C. You might have to perform initial studies to understand what are the levels of degradation that you desire to achieve. And that would entail maybe performing a study at different temperature conditions. Similarly, for high and low pH, that will entail maybe doing some scouting work at a different pH condition. Typically will be something more less harsh, as listed here, to understand that the modes of degradation that you're observing, and as again, are suitable, and now you're over degrading the molecule, or you're just obliterating the protein. And I think for the antibody drug conjugates, thing to consider is the stability of the drug linker. Being a small molecule, you may not want to stress either whether it's through pH or thermal stress for days. This may entail just a couple of hours up to a day to generate sufficient impurities to be able to assess what are the DEC pathways coming from the small molecule component. But in general, everything that we presented here, as well as what's on the paper, does also applies to non-platform biologics. But I think the thing to consider is some adaptation and obviously implementation with new analytical tools will have to be applied. So with that, maybe I can just close out the seminar. If you go to the next slide, Elaine. So here we just, hopefully we're able to convince you and present examples here as far as, what are the considerations that companies take when designing force DEC studies. Typically, this will be for the studies to support comparability studies. Those will typically be implemented late stage in development because that's when more significant process changes are implemented, right? When you're gearing towards phase 3 or commercial or launching a compound. So when that happens, obviously the risk assessment will have to be done to understand whether the inclusion of a force degradation study will supplement or will complement your comparability. And as I was mentioned, early in the presentation, there are several elements to consider. Obviously, batch release testing, characterization, as well as stability. And force DEC is just another element that will sort of will be used to boost or bolster the comparability claim. And as I said, it's typically this happens late in development when those process changes occur. So hopefully we're able to convince you that, and present here best practices, right, on how the studies are done and what stages of development. And what we're looking to hopefully gain is maybe alignment across the industry and on best practices, maybe providing some best practices guidance on that and aligning with the regulatory expectations. And as I just discussed, for non-platform biologics, we're not there yet, but I do firmly believe that we're getting there in terms of building that knowledge where some of these will actually be considered platforms in hopefully the near future. So with that, I think, Elaine, I'll hand it over to you.
Thanks, Alex. So it remains for me to thank all of the BioPhorum force degradation workstream for their enthusiastic discussions and completion of surveys and case studies, and to our fantastic authors who took the time to put all of this together into a coherent paper.